Hello guys, in this video I'm going to talk to you about the enzyme lab that we're going to do on Thursday. You're going to use oxygen sensors to measure the enzyme activity. Enzyme is derived from the um, yeast. Yeast cells actually are used as the catalyst source. So I have prepared yeast suspension here, about 14% mass per volume, and I have hydrogen peroxide here, 3%. You are going to use this container as your reaction chamber. You're going to use a stir plate to mix the yeast suspension and the hydrogen peroxide as the reaction proceeds. What else you're going to use is you're going to use oxygen sensors. We're going to use Pasco sensors. Here is the oxygen sensor in this plastic bag with label. You're going to remove it from its plastic bag. This is the oxygen sensor itself with a rubber stopper on its top, and this is another piece of oxygen sensor, and you will need an interface to connect this into your laptop because the data collection software is working on your laptop. So this is a USB link that you're going to connect to your computer, and you're going to connect your oxygen sensor into this USB link. So you connect your oxygen sensor into the USB link and then connect your USB link to your laptop, and you will see the capstone is already running on my computer, and it recognized the oxygen sensor as soon as I plugged it into my computer. As you can see, there are many choices on the first screen of the capstone. You're going to select table and graph, but there's no measurements on it. So you're going to select time for your x-axis and oxygen concentration for your y-axis in your both data table and your graph. So what are you going to do is you will place this container on top of your stir plate and use your magnetic stirrer, put it in there and start it to like a moderate stirring state. It's at 5 right now out of 10 scale in mine. You're going to measure out 10 milliliters of hydrogen peroxide using your syringe. Try to not to get it to the sides of the container so that all of the 10 milliliters of hydrogen peroxide is undergoing the reaction. And you get your yeast sample, mix it well before you place it in a container, because since it's a suspension, you want to make sure it's evenly distributed. The yeast cells are evenly distributed in the suspension. And you're going to measure 0.5 milliliters of yeast suspension, place it in the container, and wait for 1 minute to mix it up. After 1 minute has passed, you can seal the container with the oxygen sensor. I just want to make sure the yeast cells are again evenly spaced, evenly distributed within the solution, hydrogen peroxide solution. while we're waiting for the 1 minute to pass, I will show you some features of the capstone. You have your data table here. When it starts data collection, you will see it's running down here, and your data plotted on the graph right here. When it's done, you will select your data table, all the data in your data table, and you go to your graph and click on this best fit line to produce your best fit line of your plotted data. Now I'll go ahead and seal the container thoroughly and start data collection by clicking on this record button. I will collect data for 1 minute. You can collect for 2 minutes as well, or 3 minutes, whatever you want, or more, to see the reaction is how well the reaction is going. It has been 25 seconds in my case, and after 1 minute I will stop data collection and use this feature to calculate the rate of the reaction easily. But what I want you to do is get the best fit line and calculate it by hand. I mean, the software will give it to you easily, like it will calculate it for you, but it will not calculate it for you in the exam. So once you get your graph, get your best fit line using the software, and then make your calculations using your own graph. Now it's done. Right now I will remove this off, stop my stir, and clean this up. Okay guys, since you have your data already collected and plotted on this graph by Capstone software, right now only thing you're going to do is to calculate the rate for this reaction, and the conditions for this particular reaction is 3% hydrogen peroxide as a substrate, 14% yeast suspension as the catalase source, 35 degrees Celsius temperature, and pH is about 6. So you're going to change one of these conditions. I don't know what you're going to pick. You can pick temperature, substrate concentration, whatever you're going to pick. You're going to change the conditions and do the same experiment over but under different conditions. Calculate the rate for those different conditions, and then put them in a concluding data table showing you how would the reaction rate changes if you change, say, substrate concentration or temperature. So to calculate the rate in this graph, you can do the calculations with this graph, but it doesn't seem nice to me. So what I'm going to do is I will go ahead and click on this button, and then click on this sign, and it will rescale your graph starting about like 19 or 20. Okay, so you have your data plotted here, but you will need a best fit line to be able to calculate the rate. So move this around, and to be able to make the calculations of best fit line, you have to select all your data. So click on time, drag it to your oxygen concentration, and drag it all the way down. After you selected entire data for 1 minute, I have collected it 1 minute. You can do it like 2 minutes, 3 minutes, whatever. I have done it 1 minute. So I selected all my data for 1 minute, and I will go ahead and here select linear fit from this drop down menu. When I hit linear fit, it will show me this best fit line for this particular data collection. And it will also give me this these statistics, your slope of this line already calculated, and your regression, your R value. Now you can move this around to be able to see the data plotted on the graph better. And I want you to make your calculations using this line by hand. Okay, so you're going to select 2 points on this line. Say one of the point is this, the other point is this. So X value for this point is 50, Y value is whatever, it's like 23.7 or so. Okay, so these are your X2 and Y2 values. And select one X1 and Y1 values. Say it's 10 and 21.2. And then make your calculations. Simply, the slope of this line is y2 minus y1 divided by x2 minus x1. And record your calculations as the slope of this line percent per second. Okay, your oxygen concentration is percent per second. You could change this into parts per million too, but it's percent, since we have started it as percent. Now you can't change it, but you could change it in the beginning of the experiment by just selecting it as parts per million. So that's everything you have to do for this experiment. You're going to do the same thing for all of your different conditions. If you're doing temperature for 10 degrees Celsius, 20 degrees Celsius, 30 degrees Celsius, you are going to do the same experiment and calculate the rate for each of those. And then put those in another data table telling me that I have tried this experiment 10 degrees Celsius and the rate was this. I've tried this in 20 degrees Celsius and the rate was this. And plot that data into another graph, concluding what would happen if you change the temperature, what would happen to the rate of this reaction. All right, I hope this will help with your experiment, and see you on Thursday with our lab.